Kalau komenoni barang phone charge नहीं करूँगा
Spot negative.
नहीं charge कर
I think it's long charge.
अलग है नहीं कर सकते सही है
No, it's in the charge.
charge ना दे देखो देखो नहीं करेगा नहीं power bank के
Plug base है ना?
plus base है
Oh, no.
make it home नहीं right charge नहीं करती नहीं थी ज़्यादा बड़ा करेंगे
No, no. Well, then Mama da. Sit there.
वो order नहीं है ना
Sir, washroom में खोल के.
हाँ है
Okay. Good evening. Can you hear me? Can you hear me and see me clearly? Let's start shortly. Yeah. So it's 8:09. Let's wait a few minutes and we can start the discussion. So, yeah. Can you hear me? Is there a lag or all good? Right. Okay. Right.
Recording in progress.
So I think we better start. So this is our first discussion. So this is happening as the first discussion in my 10-month program for 2026, right? So your exam is going to be held in August in 2026. So I started this plan so that I can cover the syllabus as sort of a rapid theory and also cover the previous grade 12, particularly grade 12 lessons as a rapid revision. At the same time, we are doing these sort of paper sessions, right? So since this is our first session, it will be just your own self-marking. You are going to write it yourself, and it's going to be your self-marking. But from next month onwards, so next month also we are going to have a even bigger and also complete paper. So that paper will be marked and sort of given to you with your own, right? So those kind of exams will start from next month. And yeah, so this is our first session. So it's sort of the start into our this program. That's why I just gave it as your own self-marking and self-writing. So the discussion I'll be uploading for the people who couldn't join live, right? Do you have any questions regarding the session that before we start? Everyone got the paper, right? Everyone got the paper. Hands up anyone who's from ISM? Got the paper from ISM? Got a hard copy from ISM? Anyone? Hands up. Okay. This, I think you all have soft copy, the PDF. Anyway, so I hope you have gone to at least just even through the paper. So because it's a session and also it's a free session, I limited it to a few questions, right? A few MCQs about 25 to 27 mark, right? And then I included 3 essay questions. I'm going to discuss the whole paper. So discuss the whole paper. I have given a few essay questions as well as, so most of these questions I targeted the grade 12 syllabus because in the rapid theory we just started with reproduction, right? We started with reproduction and we are going forward as well as the grade 13 entire content as rapid theory, as well as at the same time from next month onwards I'm going to start grade 12 rapid revision. That is going to be a revision, right? Just going over the content that is related to you to go and answer the paper, right? So that's going to be a rapid revision, but the grade 13 entire syllabus, it's going to be repeat, I mean, as a theory class, but in a sort of a fast manner so that I guarantee the syllabus coverage within the 10 months, not 10 months anymore, about 9 months and a week that you have left, right? So that is the plan, okay? So that's why I limited this paper to the grade 12 syllabus. Okay, so without further ado, I think let's start with the essay discussion. Okay, let's start with the essay paper. Before we start, anyone having any connection issues? Can you hear me clearly? Anyone having any connection issues? Can you hear me clearly and see me? All good. Right. Anyone who doesn't have the paper? Anyone who doesn't have the paper? No one. Okay. So let's start with our short note question, right? Let's start with our short question because I feel like I don't know if it's applicable to everyone, right? I feel like most of the people are not familiar with the exact art of writing a short note, even at this stage, right? Exact art of writing a short note, even at this stage. So, what are the things you have to keep in mind when you're writing a short note? Right? As you know, you are going to do the short as sort of your last, if you are running out of time, or if you didn't have any confident questions as the question number 5, 6, 7, or 8, or 9, if you didn't have any confident 4 questions among these, you are going to jump to the short. That's the situation, right? But I recommend that you give the short note a try, right? Because one thing, it saves time, right? It's time saving, right? And the second thing is there's a reduced risk of getting it completely wrong. Do you get it? Right? So if you sort of miss the exact point what they're asking, for example, they're asking the histological structure, right? So they are being very specific, right? But if you miss the exact point, or let's say the objective of an essay question like this one, this liver question, you are going to lose a lot of marks within that essay question. But in a short note, because you have 3 parts, there's a reduced risk of losing a lot of marks, because if you sort of misinterpret one is short note, there's a chance of getting the next, I mean, the remaining two to a certain extent. Right? And also because they are asking the short note questions from like several places of the syllabus, there's a chance that you are at least able to answer about 2 questions of the 3 essays, right? 3 short notes. Okay, so that's why. So there's a reduced risk. There's a reduced risk with considering short notes as well, right? So those are sort of why I recommend, right? If you're running out of time after answering 3 essay questions, give the short note a try, right? So what are the things you have be mindful? What are the things you have to be mindful when you are uh, sort of starting to write the short note? What are the things? Right? Short notes you can write in point. Short notes you can write in. You can use point form. What do you mean by point? The thing is, you don't need to write complete sentences. You don't need to write complete sentences, right? So I'll go through these short note questions very shortly. But before going to that, I'm going to give you these points, right? You don't need to write exact sentences, but according to your marking schemes and also the standard of the marking, you cannot put bullets. May bullets down there. Star marks down there. You can't put star marks, right? You can't do any of those things, but you can write in point form, sentence by sentence. You can write in point. That's the benefit, right? So I know most of you are studying in point. You are studying in point. මතක තියාගන්න after sentence by sentence as a point, right? So you can go and exactly write in that form. Got in the form again, you can go and write the show. Okay, so those are the benefits, right? So let's move on to the short note questions that we have, right? Let's start with this one: effects of smoking. Okay? Effects of smoking. So I hope everyone has gas exchange, right? At this point, I don't know where you are in the sort of the theory class, right? But at this time of your A-level career, you should have, I mean, at least you are, I think, I don't know, where are you exactly in your sort of normal theory class? Where are you? When you last into the theory class, where were you in the theory class? What was the last lesson? Anyone? Yeah. Where are you in your normal theory class at this point? What is the last lesson you discussed? Anyone? Immunity? Okay. Immunity. Right? Okay. Anyone who's completed with the grade 12 syllabus, it's done with excretion and everything? Okay. And respiration. Okay. So That's why sort of I started this 10-month program, because as you can see, you should be able to figure out now. I mean, most of you should be able to figure out now, according to the way you are sort of, I mean, the pattern is going. There's a chance you might not be able to cover the syllabus, right? So that's why I started this rapid theory and rapid revision, sort of all hybrid class with the 10 month plan, right? So anyway, moving on to this short note, what are the things you have to mention, right? I'll just underline the points, right? I'll just underline and give you marks, right? Cigarette smoke harms nearly every organ in the body, including the organs of the respiratory system, and increase the risk of illness, disability, and death, right? Smokers inhale a large number of chemicals which mainly come from burning tobacco, right? So I think the first point should be harms nearly every organ in the body, that is one mark. Increase the risk of illness, disability, and death, that is the next point, right? You have to say, right? You have to start with the introduction. So it nearly harms every organ, particularly the respiratory system. I mean, that is included in the idea. They inhale a large number of chemicals which mainly come from burning tobacco. Dumkola kuchala in a smoke, right? A cigarette is what? That is coming from the burning of small pieces of tobacco leaf, right? Most of these inhaled compounds are chemically active and can trigger damaging changes in the body, right? Okay. So, we have a list of active components in the cigarette smoke that you have to know what is the effect and what is the damage. You have to know two things. You have to know two things about each substance, right? You have to know what is the effect on your body, and what is the damage cause because of it. Those are the two things. You know what is the effect, what is the damage. So if you take nicotine as the example, so that's one mark mentioning nicotine. Yeah. It is addictive, you have to mention. It is why people smoke. It is why people smoke, right? So you have to mention it is an addictive drug. Temporarily increases the harm. That is the effect. It is also an effect. Okay. The constriction of peripheral nerves. It is again seeing a temporary increase in blood. Actually, nicotine, other than the fact of being addictive, it is actually having very sort of, let's say, low harmful impacts. But because of the nicotine, people smoke. And because people smoke, they also inhale the other chemicals. Because of these other chemicals, they get all these problems. Right? So, they say cigarette smoke as a whole, right? Next point, simulates the secretion of mucus by the goblet cells. One mark, right? And also inhibits the action of cilia. 2 points. Causing accumulation of mucus in the bronchioles and accumulation of mucus in the bronchioles and blocks. What are the bronchioles? Right? So, in your respiratory system, right, you have the trachea and it divides into the left and right main mucus, and the left and right divides into bronchioles. Okay? Small and small. Tubes delivering air in the end to what? To the gas exchange surface, that is to the gas exchange surface, that is the alveoli. Right? So your alveoli, no, sorry, your bronchioles particularly are composed of smooth muscles. Right? They are composed of smooth muscles. They are component, I mean, the amount of cartilage. Right? So in your trachea, right? This is the sort of, here you can feel your trachea. Here you can feel your trachea. That is mainly, I mean, it is sort of strengthened by cartilage. The cartilage rings to the trachea. So it is cartilage. But the amount of cartilage decreases as you go down the respiratory system. amount of cartilage decreases. Okay, and more smooth muscles. Smooth muscle are susceptible to what? Inflammation. That is the next point, right? These cigarette smoke can inflame, right? Can cause inflammatory changes. What is inflammation? What is inflammation? It's a local reaction to something irritating. It's a local reaction to something irritating, right? So this cigarette smoke can cause inflammation, and so any inflammation, what do we call? We call any inflammation as what? Itis. We call any inflammation as itis. If your pancreas is inflamed, pancreatitis, right? If your liver is inflamed, we call it hepatitis. What else? Bronchioles inflammation, bronchitis. Your sinuses inflamed. Sinuses are located where? In your head, in your frontal lobes, you have sinuses. If your sinuses, these air cavities in your skull, if they are inflamed, sinusitis. Right? Any inflammation is itis. Okay. So bronchial inflammation is caused by cigarettes, causing bronchitis. As a result, breathing may become difficult. That is the next. Then that is the next chemical that you need to know. Hydrogen cyanide. Right? Stops the cilia from working properly. That is the previous point. Due to the loss of action of cilia, dust and other particulate matter get collected. Dust and other particulate matter is collected, increasing phagocytic cells. What are phagocytic cells? Anyone? What are phagocytic cells? What are phagocytic cells? Phagocytic cells. What do they do? If you have done immunity, phagocytes, what do they do? They are what we call as macro. Right? So phagocytic cells, what is the main phagocytic cell in your lung? They are macrophages. Macrophages do what? Phagocytosis. Phagocytosis is engulf foreign matter. It could be foreign means, it could be other bacteria, particulate matter, right? In this case, dust, right? So the main phagocytic cell in your lungs are your macrophages, and they can sort of eat these particulate, right? That eating can release lytic enzymes, right? So that eating action can release lytic enzymes, and these lytic enzymes can pose as a byproduct, right? As a byproduct. So lytic enzyme doesn't damage only these. I'm going to engulf only these foreign particles, right? Because it's an enzyme. It's going to damage the surrounding tissues as surrounding tissues get damaged, right? Because of that, the alveolar tissue is destroyed. And what happens when the alveolar are destroyed? The surface area available, effective surface area available for gas exchange decreases, right? Then what is the next component? Carbon monoxide. Carbon monoxide is what we call as a respiratory toxin. Carbon monoxide is respiratory toxin. Even if you remember the reaction, carbon monoxide plus yes, irreversible. Anyone know what is the compound formed by oxide and carbon monoxide and hemoglobin receptor modified? Anyone? It's not in your syllabus. It's called carboxy. I just write HB, okay? Carboxyhemoglobin. Carboxyhemoglobin. Carboxyhemoglobin, if you can see, I drew an arrow like this. I didn't draw an arrow like this. If I drew an arrow, if I drew an arrow like this, what does it mean? It's reversible. But I drew is an arrow like this. This means this is irreversible. What do you mean irreversible? That means this hemoglobin is cancer. This hemoglobin, you can't use it because once carbon monoxide has combined with hemoglobin, that hemoglobin is useless. That hemoglobin is useless. That's why we say it binds to hemoglobin better than oxygen. And no, that is not the point. I'll give marks. I'll give marks to combines irreversibly with hemoglobin. Decreases the amounts of oxyhemoglobin produced. Therefore, oxygen transport is decreased. Right? That's clear. That is about carbon monoxide. Then carcinogen, very important. Today I talk about carcinogen as well. Contains large number of cancer-causing substances, also called as carcinogens. 90% of lung cancers are carcinogen. I'll show you some statistics. Lung cancer statistics for your interest, right? Long-term exposure to such chemicals resulting in the proliferation of cells in the bronchial epithelium, forming a mass of abnormal cells. The cancer may develop among these cells. If the cancer cells break free, cancer may spread to other parts of the lung and to other organs. I have written the right, so I was going to ask, what do you call when an abnormal cell that is at an original location and spreads to a far away location? What do you call? Metastasis, right? Metastasis. I like a bit more. Okay, you can send messages to the chat. Okay, right? It's called metastasis. So metastasis can happen. Okay, I'll show you some diagrams with. I'm not going to teach you about this other or respiratory disorder, but I'll show you right about asbestosis and silicosis because I don't have time tonight, right? But I'll be revising these sections again, right? I don't know if you can see clearly, right? It's loading, so give it a minute. Right? Can you see this diagram? This is what we call as silicosis, right? This is a lung with silicosis, right? Can you see these white areas, right? These sort of glistening areas. These are parts of the lung having silica particles. Silica in the silica. It's a mineral. Silica is a mineral, right? When these people inhale silica for a long time, right? It can cause this sort of deposits, silica deposits, right? And this is how you can see these deposits in an X-ray. Can you see here? Right? It's, after they are deposited, you can see this sort of what we call as calcification, right? There are calcium deposition around it, right? I'm going to do this, right? So this is silicosis plus tuberculosis, right? This is a real lung. It's a diagram of a real lung, right? So you can see some silica deposits in these areas. I don't know if it's that clear to you, right? So you can see silica deposits in these sort of areas, and you can see yellow color areas in sort of these places. That is because of the tuberculosis infection, right? So read about tuberculosis in the resource, right? Tonight I'm not going to talk. It's an infection of the lung by a bacteria, right? Mycobacterium tuberculosis. Okay, so I'm just for your interest, I'm quickly showing you this. This is due to this is I think because of silicosis also, right? Still, this is silicosis. Can you see the damage caused to the lung, right? This is can you see the damage caused to the lung? It's called as a honey, right? Honeycomb. Honeycomb, right? Okay. This is when some lungs damaged, right? Okay, some statistics I can show you, right? Just among the national cancer registry, sorry, right? In the national cancer registry among males, right? So leading cancers among males, you can see this graph, right? So other cancers means the total of all the other cancers not included here, right? Other cancers can occur in almost everywhere, right? Pancreatic cancer, other, right? Can you see most of it is oral cancer. In Sri Lanka, most of the males are getting oral cancer. What is oral cancer? Cancer in the mouth. Cancer in the mouth. Oral cancer. That is because of why? My video and voice is lag. Still, is it there? Are you sure it's my connection? Is it anyone else having the same issue? Anyone else? It's, now okay. Anyone else experiencing this same issue? Yes? No? Right? Yeah. Okay, let me know. So, as I was saying, oral cancer is the most common cancer among males. This is some for your additional knowledge, right? We'll quickly move on to the discussion quickly, right? This is because of what? Chewing our bullet beetle, beetle leaf together with areca nut and all this, and that. We even had calcium carbonate, lime, we had calcium bicarbonate. We had all those things. This is what I wanted to have your interest because I'm talking about lung diseases. Trachea, bronchial, and lung cancers are the next. That is because of smoke. That is because of the smoke. Colorectal means in your colon. Colon is part of what? Anyone? Colon is part of what? Colon. It's part of the colon is part of the large intestine, right? Right? Yes, colon is part of the large intestine. So colon and the terminal part of the large intestine, that is a rectum, very common. That is also associated with smoking. That is also associated with smoking. Colorectal cancer has a very high connection, very close connection with smoking. Then esophageal cancer also very close connection with smoking. Esophagus is what? Your food tube, right? There is also, then prostate cancer is mainly genetic, right? Prostate cancer is mainly pharynx, those are upper respiratory. Stomach and bladder is also closely related. These are female cancers, right? Female cancers, you can see most of the females are not smoking, right? So I will not add it. Then breast cancers, right? As a genetically, most of it is genetic breast cancer and thyroid cancer, cervix cancer, right? Right? Those things can happen, right? So quickly, I just want to show you how cancer progresses, right? So carcinogens, right? We can call it how cancer, lung cancer origin. So I'm just quickly teaching a theory part. Right? Common carcinogens are included in the smoke, cigarette smoke, industrial hazards such as silica. What else? What else? The industrial hazards, asbestos, asbestos. Any other? So silica dust includes other sort of sort of. Okay. Then air pollution. Air contains carcinogens such as these sort of reactive oxidizing gases, right? Reactive gases, right? Then genetic component, right? Genetics as an issue. like strong influence. So this is what a normal sort of respiratory epithelium is supposed to look like. It might not be that clear. You can see the cilia. You can see the cilia, right? So now this is the beginning of a cancer, right? But I'll quickly jump to the end. Can you see? There's a lot of cells, right? There's a lot of outer small cells. Yeah, Kissima pilula. Kissima pilula. There is no orderness. There's no order. There's no proper arrangement, right? So this is the end stage, right? Can you see the difference? Nicely arranged cell layers. Nicely arranged cell layers here and here. Complete mess, chaos, right? So those are the differences, right? So this is again a sort of a tumor in this stage, and this is, can you see this diagram? This is lung metastasis. Lung metastasis, right? Can you see lung metastasis? There are abnormal cell masses everywhere in the lung. This lung is completely useless. This lung is completely useless, right? Everywhere you have abnormal cell masses, right? This is sort of the early stage. Here also you can see tumors, right? Okay, so that's what I sort of wanted to explain about that short, right? So that is that theory. Okay, clear about that short note? Next, let's move on to the other short note. I think the second short note is the rules of nomenclature. Rules of nomenclature, right? So I'll refer to the resource book for that. Right? Where do we have rules of nomenclature? Evolution. So I like to refer the resource book whenever discussing an essay. I like to refer because why? Even when I'm discussing, I'm trying to familiarize with the resource book because resource book is the key, right? With the resource book is your Bible, resource book is your law, right? Right. So it's your, until you do A levels, it's your political. Whatever it says, sometimes it's wrong, but sometimes it's controversial. But you have to study that and go on. So the key to that is whenever I'm teaching also, I like to refer to the resource book because even then I'm familiarizing yourself with the resource book, right? So what are the points you have mentioned about rules of nomenclature, right? People who just joined, we are discussing the essay part first, the paper, essay questions first. Okay. What are the things you have to mention, right? Rules of nomenclature. Two species cannot have the same name, right? I'll give you 2 marks. Each species has a generic name and a specific epithet, together forming the scientific, right? 4 marks. Name should be made up of Latinized words, written in Roman script, 2 marks. Should be underlined when handwritten, italicized when printed. Okay. First letter of the generic name must be capitalized. Specific epithet must be in simple letters. Pretty straightforward, very important otherwise, right? I think you can remember the theories. Ignore this. In scientific writing, the name of the author who gave the name can be indicated by a capital letter, right? Ignore this diagram, I can't use it. Okay, right? The previous annotation, I have forgot to erase it before putting it on the board, right? So you can see the example, Cocos nucifera, you can see the first letter is capital and all the other letters are simple, and Carolus Linnaeus gave it. So we are sort of paying tribute by writing his name as an abbreviation capital letter, right? Subspecies we can indicate by the third word, right? So Panthera pardus. So we have the species name and the specific epithet by Panthera pardus. But Kotia is sort of mentioning that it's endemic to Sri Lanka, right? So that's a subspecies of leopard, Sri Lankan leopard, right? There's another example I can give. The Sri Lankan okra. Jam. Again we say Naja. Right? So I or this I hand wrote this. I write it. The name of Sri Lankan okra. Okra dano Nalaya. Right? The scientific name of Sri Lanka is Naja. 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 Okay. Right? Okay, you go and spell it and see. Right? So you have to underline. So this is the subspecies, the third Naja. Right? It's the third word is indicating its subspecies. Right? So those are the main applications. Okay? So those are the points. So the third word can be used to represent a subspecies and other points. Right? So that is the second short note question. Any questions from the second question? No. You can ask any question. Okay. Then first short notes are from down to top. I like the last topic, so I started with that. So people are interested. Okay. Then effect of temperature on enzymatic activity. Right. Effect of temperature. One of the things you have to mention. A bit of physics and chemistry you have to say. Temperature increases kinetic energy, right? So therefore, the molecular motion increases. Increasing temperature increases molecular motion. Speed of the moving molecules you have to mention both enzyme as well as substrate will be increased, right? So if this is the enzyme and this is the corresponding substrate, both of these will start moving very fast. Right? Speed will increase. That is because of the increased kinetic energy. So you have to mention not only the enzyme or substrate, but both. This will enhance colliding probability. For what? නිකන් මේ this substrate ඇවිල්ලා මෙහි දුන්නට වැඩක් මෙහි කොහෙවත් දුන්නට වැඩක් where does it have to go and collide? නිකම්ම collide වෙලා it has to collide in a orientation exactly at the active site. It has to collide with the active site, right? Therefore, the colliding probability will increase because of this increase, right? Collision between enzyme active sites and substrate. Greater chances here. This continues up to a certain temperature. Continue up to a certain temperature, you have to say. Then there is a rapid decline. This temperature is covered by the okay. This may vary from organism. So our body temperature is what? We are warm-blooded animals. So we have to maintain our body temperature between really, right? Our body temperature is what? 37 to 39 is the range. Right? So this means you are feeble, right? Okay. So our enzymes will have to be maintained in this temperature range. Because why? It's their optimum temperature. It is the temperature where they are functioning in their most efficient. Right? It is their most efficient, that temperature. That's why we are maintaining the temperature. Okay. So hot springs bacteria. Give me a name to all these bacteria. They like hot temperature. So we can call them thermophilic. We'll learn about these different kinds of bacteria when we are doing microbiology. These are thermophilic bacteria. So they like temperature. They live in hot temperature. Therefore, their enzymes at optimum temperature. Now we have mentioned. So I want you to mention the two examples: human enzymes and these thermophilic bacteria enzymes. We have to mention those two examples. When the temperature increases beyond the optimum temperature, what are the ones that are disrupted? At least two, right? So they are given two. So for the two, you are going to give one. Hydrogen moves and ionic moves may get disrupted. This will result in a change of the shape. So change in the shape of the active site one part. You have to say, which will alter the complementary nature. Therefore, the complementary binding you don't have. This event is called denaturation, enzyme denaturation. Therefore, the rate will decrease when the temperature increases beyond the optimum temperature, although the rate of collision will keep on increasing. Okay. Any questions? These are things you learned in grade 12 in the first few months, right? So this is your optimum temperature, and you can see gradual decrease, but the rate of reaction, does it become zero? The methanol rate of reaction has become zero. Just a quick thing, just a quick bit of logic. Anyone tell me? Yes. After a certain temperature, does the rate of reaction become zero? Something logical? Chemistry knowledge? Yeah. Does the enzyme, sorry, the rate of reaction becomes zero? Does the rate of reaction become zero? No. Why? Because even though, so now after here, let's say after about 50 degrees Celsius, there are no enzymes, right? There are no enzymes. But do we need like univariate enzymes, or do we necessarily need enzymes? Right? So still the products, sorry, the reactants can still become products without enzymes. Enzymes are just there to do what? Just to catalyze. They are just catalyze, right? Enzymes if you inactive, say the reaction is not happening. So actually this graph is a bit, right? The rate does not become zero, right? The rate does not become zero. With the rate, sorry, the reaction very low rate because enzymes are not. This is the logic. Okay. So this is our sort of the final short note question. These are the points I want to write in the short. Any questions? Can you clearly hear me and see? If it becomes zero, yes, so that means your temperature has increased to an intolerable temperature. So definitely close to 60 we can close to 60 temperature, body temperature. Sorry, close to 60 degree body temperature we cannot suffer. Okay. Alright, because mainly because of these erratic reactions, our sensitivity brain can be damaged because of the hot blood, because of the warm blood circulating through the brain, our neurons become damaged, right? So that is the beginning, and the heart becomes very affected because of the high temperature. That's the cause of dying, right? Maybe. Okay, any questions? Let's move on and do some MCQs, right? Okay, so let's start the discussion of MCQs, right? Otherwise, it's getting late. Okay. So first, let's do some MCQs and come back to the essays, right? Otherwise, boring. Direct, straight answer MCQs right? Which of the following organisms in plant cells convert fatty acid into sugars? Fatty acid into sugars. What's the answer? Specialized peroxisomes found only in plants. Yes, specialized peroxisomes found only in plants. Glyoxysomes convert fatty acid into sugars. Simple things, right? Simple, direct MCQ points you have to study and remember, right? Lysosomes have what? They have what we call as hydrolytic. Hydrolytic. Peroxisomes, what sort of enzyme do they have? They are oxidized. They break down peroxides. Yes. Endoplasmic reticulum is acting as most of it knows, so it is in receiving, packaging, and redistribution, those things. Sorry, yeah, I was sorry. Endoplasmic reticulum has so many functions. Functions as the membrane factory, production of carbon in carbohydrate metabolism, right? Storage of calcium, blah blah. We have a lot of endoplasmic reticular functions. Golgi is your host of this. Okay, so answer number 3. Next question, correct regarding prokaryotic organisms. Prokaryotes. What do you know about prokaryotic organisms? First statement, all prokaryotic organisms are heterotrophic. What do you mean by heterotrophic? What is the opposite of heterotrophic? What is the opposite of heterotrophic? What is the opposite of being heterotrophic? This autotrophic. No? Autotrophic means what? What do you mean by trophic? It refers to their carbon source. Carbon source. Okay. Autotrophic organisms can fix inorganic carbon. What is your inorganic source? What is your source? Carbon dioxide. They can fix them into organic carbon. Heterotrophic people cannot fix. Heterotrophics cannot fix inorganic carbon, so they have to use organic carbon. All prokaryotic are heterotrophic. There are. Give me an example for prokaryote that is that they can fix their own carbon. Cyanobacteria. They are photosynthetic, so they can fix their own carbon. So no issue. You can't say. Second one, all prokaryotic organisms do not have peptidoglycan in their cell walls. Do not. Yes. How much prokaryotes? Other prokaryotes that we have? Bacteria and archibacteria. Bacteria and archibacteria. Right? Peptidoglycan in domain bacteria. In domain archaea, what do you have? Archaea, how do you spell? Kaya, right? Several vowels in there. Domain here. Domain archaea. What do you have? Cell wall. Cell wall is composed of what is the right answer? Domain here. What is the correct answer? Protein. Yes. So, what does that mean? The statement is sorry. The statement is correct. Right? They do all of them do not have a right? Yes. They can have other things. That's what it is. All prokaryotic organisms do not have sorry, all prokaryotic organisms can fix atmospheric nitrogen. All can't. Some can. Welcome. You can correct this by saying some. There are nitrogen fixing prokaryotes, nitrobacter, nitrosomonas. Most people can be. All prokaryotic organisms do not have ribosomes. Why? Having ribosomes is a feature of all living cells. Having ribosomes is a feature of all living cells. So, any living cell, if you take, they have ribosomes. So, you can't take this. All prokaryotic organisms are not microorganisms, they are all microorganisms. There are no prokaryotic organisms. All prokaryotic organisms are not microorganisms. What do you think? They are all microorganisms. They are lagging also. They are all microorganisms, right? So there are no, I mean, there are no prokaryotes, there are not microorganisms. Microorganisms are no one prokaryotes la kamu. Got it? So yes, they are all microorganisms. So you can't say not. So that is wrong. So what is the answer? Answer number 2, right? Answer number 2, right? Right? Any questions? Moving on. Third one. The base adenine A of an organism, so of DNA of an organism form 23.3 of its composition. Which of the following is the likely base composition? So adenine, they say 23.3, right? Adenine is 23.3. So what is the base pair rule? We have to apply that. Adenine binds with 2 hydrogen bonds with thymine. Guanine, 3 hydrogen bonds with cytosine. So you can apply this gas 23.3% adenine. So what can you directly see? Adenine තියෙන තරමටම thymine තියෙන. Adenine තියෙන තරමටම thymine තියෙන. So AT 23.3 is a correct statement. So we have 3 statements in it. Then what do we have? 22 3.3 times 2 is 46.6 net. 100 within make up කරනවා. What do you get? You get 60 what do you get? 63.7. So you divide this by 2, right? Not 7, no? So we are doing 146.6 from here. 63.4. Yeah. So 63.4 divided by 2 is how much? 26.7 rate of net. Right? What is the answer? People, what is the answer? Round 3. Eh? TC ratio is equal. Round 3, right? Okay, clear. What answer did you get? Okay. Moving on. Which of the following structure function combination is incorrect? Incorrect, right? Nucleolus ribosome production. Nucleolus located within the nucleus. nucleolus, nucleus is a double membrane bound structure, right? Double membrane bound structure. There are openings. What do we call these openings? What do you call these openings? Nuclear pores, right? Nuclear pores. So, now what else do we have? Sorry, membrane structure. Inside, you have chromatin, right? Chromatin fibers you have, right? And other than that, we have a densely stained area. Densely stained area. I mean, in electron micrographs, we have this densely stained area, right? That is for the nucleus. What are the functions of the nucleus? What are the functions of the nucleus? It is the source of what? Ribosomal. It is source of RNA. So, yes, you can say nucleolus is involved in ribosome productions. Glyoxysomes photorespiration in plants. Photorespiration theory. There's a wasteful process happening in plants that is called photorespiration that produces toxic products. A toxins, what do we have to do? We have to metabolize in a pathway that is called photorespiration. What are the organelles that you need for photorespiration? Peroxisomes. Then mitochondria. Not glyoxysomes. You don't need glyoxysomes. You need peroxisomes. Cytoskeleton determines the shape of the cell, yes. Particularly in animal cells, cytoskeleton provides strength and maintains the shape. Vegetables store soluble substances needed for cellular activities, yes. Sugars, pigment molecules, right? All those things are located in the vacuole. Golgi complex produces lysosomes, yes. Golgi is the source of lysosomes, right? So, what is the answer? Second. Any questions you have, you can ask, right? Can you clearly hear me and see me? Any issues? Okay. Next question. Which of the following is common to lactic acid fermentation, ethyl alcohol fermentation, and aerobic respiration? So we have two forms of anaerobic and the aerobic respiration we have a right? Aerobic means what? There is no oxygen. There is no oxygen. So what is the function of oxygen in respiration? What is the function of oxygen in respiration? It is what we call as yes, final electron accept. Oxygen is the final electron acceptor. So oxygen becomes water in cellular respiration. Sorry, aerobic respiration. It is water. That is if there is oxygen. This happens. What? The linking reaction, Krebs cycle, electron transport chain. In the mitochondria. But if the mitochondrial reactions only happen if there is the oxygen only one reaction happens. What is the only reaction happening without oxygen? Glycolysis. Nothing else happens. Without oxygen, nothing else happens. Without oxygen, nothing else happens. Simple question, right? Quick reminder. Comparison between C3 and C4 is incorrect. We ask the incorrect statement. C3, CO2 fixation occurs only once. Yes, where? Rubisco vitarayine. Rubisco is involved in CO2 fixation. CO2 fixation occurs twice? Yes. What is the other CO2 acceptor? What is the other CO2 acceptor? Phosphoenol pyruvate. Phosphoenol pyruvate is the CO2 acceptor in where? Mesophyll cells. In mesophyll cells. So yes, CO2 fixation occurs twice, yes. Major CO2 acceptor is RUBP, yes. Major CO2 acceptor is phosphoenol pyruvate, yes. Sorry, here it's not visco, right? So visco is the enzyme. So CO2 acceptor is actually the CO2 acceptor is what? RUBP, right? Ribulose 1,5 viscose. Third statement, CO2 fixing enzyme is RUBP carboxylase in C3. No, no, RUBP carboxylase is RUBP carboxylase oxygenase. Very simple, right? RUBP carboxylase oxygenase is another name for Rubisco. So that is correct. CO2 fixing enzyme is PEP carboxylase, that is also correct. What is the reaction? Quick reminder. PEP plus CO2 produces what? Anyone? Phosphoenol pyruvate plus produces what? In C4 plants. Only 9 more months left. Okay, you can buy a current on it. Right? You have to be in touch. Be in touch. I'm going to revise these sections again. Right? But you have to know where you are, let's say, not your knowledge, but you are sort of, let's say, keeping in touch with the syllabus, right? You have to keep in touch with the syllabus, right? You might have understood at a certain point, but now you might have. So that's the point. Produces oxaloacetate. Oxaloacetate is a 4 carbon C4 path. That's why we call it C4 path. Reaction is catalyzed. Photosynthetic yield is high now, usually because of the inefficient process of photorespiration. C3 plants have reduced efficiency yield, but in C4 there's no such problem, so yield is usually high. First product of photosynthesis is PGA. First product of photosynthesis of cells, yes. So you know CO2 plus RUBP in the Calvin cycle. CO2 plus RUBP in the Calvin cycle. This is what 6 carbon unstable intermediate. It becomes 3 PGA or just PGA. This is the first reaction of the Calvin cycle. Carbon fixation. Okay. So that is correct. So what are they asking? Incorrect set. Statement number 4 is MCQ number 7. Key characteristic of each of the organisms labeled as ABCDs as follows. Right. So bit of plants now. We are jumping to some plants. Right. What is the answer you got? What is the answer? What is the answer? Heterosporous. I think we have to go through the given answers. Let's consider the first answers. Heterosporous, we have cycus. Heterospore means what? There's a macrospore and a megaspore and microspore. Megaspore, microspore. Well, the heterosporous, right? So cycus has this mango. I mean, finals, let's keep it because it's not in your syllabus. Let's see if we can get an answer by things you know, right? Okay. Mango? Yes, because mango is a flowering plant. Heterosporous is an evolutionary advanced feature. Being heterosporous is an evolutionary advanced feature. So definitely mango is there. Banana is also a flowering plant. Yes. Pogonatum. Pogonatum is homo. It's homosexual. Right? Ikajati is poeta. Right? Pogonatum is homosexual. Right? So we have three answers possible, but let's see, keep two in mind as well. Photosynthetic gametophyte. Photosynthetic gametophyte, you can find it one non-vascular seedless plants. In a primitive mind, Pogonatum like again, right? So Mercantia, you can take, right? Pogonatum, you can. Nephrolepis, what do you think about Nephrolepis gametophyte? Do you remember? Can you remember green color? Sorry. Sometimes it's very okay. Can you remember this sort of heart-shaped photosynthetic cells with some rhizoids, right? And this is what? Nephrolepis gametophyte. So, yes, Nephrolepis gametophyte is photosynthetic. You can still take that. Cycus, let's keep minus for back because I took this from my old syllabus page. Cycus, definitely not photosynthetic. Gametophyte is not photosynthetic because very, very reduced gametophyte. Right? So you can't take cycus. So we still have these answers to consider. Seedless. Who are the seedless people? Selaginella, very definitely seedless, no? So Selaginella, does it produce seeds? No. So you can take. Cycus produces seeds. Yes. So you can't take. So who is out? Cycus does not produce seeds. Lycopodium, seedless. Yes, you can take. Nephrolepis, seedless. You can take. Right? So answer number no. Thalloid gametophyte. Gametophyte is thalloid gametophyte. You can find only the thalloid gametophyte. Yes. Yeah. Next answer is answer number 3. No? Mercantia. Right? So mango is heterosporous. Nephrolepis gametophyte is synthetic. Lycopodium is seedless. Mercantia is having a thalloid gametophyte. Any questions? Any doubts? Okay, shall we move on? In the classification of organisms, the taxon phylum was introduced by who? What's the answer? So I want now answers. Taxon phylum was introduced by Dynasty. Dynasty. Right? These are direct MCQ points. You have to be familiar. Right? Dynasty. Right? So moving on. Phyla that includes organisms having glycogen as their main stored food. Glycogen. Fungi, And animals, fungi and animals have glycogen, no? Right? But here only a bit of some, so Pitidiomycota lycophyte, right? So those are, this is a fungus, right? So you can take that. Pterophyta and zygomite, this is a plant, no? So you can't take lycophyta, you can't take. Chordata, yes, animals, and chrysophyte. Can you take chrysophyte? Chrysophytes, are they, what are they, chrysophytes? Chrysophytes? These are, we call as, it's a type of algae, right? No, sorry, not chrysophytes. Sorry, these are, yes, algae, but we call them as chrysophytes are diatoms. Diatoms. Cycadophyta and phaeophyta. Cycadophyta means what? They are plants, so you can't take. Phaeophyta means again type of algae. Basidiomycota. Oh my, both are right. Basidiomycota and ascomycota. Both are fungi. Which of the following structures can be seen in both annelids as well as arthropods? Annelids as well as arthropods. There are some features that are unique to, there are some features that are unique to annelids. Titellum parapodia, those are unique. So definitely you can't take. Capillaries means what? Let's forget ventral nerve, right? Capillaries means what? In order to have a capillaries, you have to have equal as closed circulatory system. Closed circulatory system. Right? You have to have a closed circulatory system. So out of these, does arthropods have a closed circulatory system? No, they have open circulatory system. They have hemolymph. They don't have blood, right? They have hemolymph. So that is incorrect. You cannot. You only arthropods really. Annelids have capillaries. Chitinous exoskeleton you can only see in arthropods. So ventral nerve is something common to nature. So let's see. I think I included some items for this comparison. We went through this. Right. You can see a bit of annelid structure. Right. You can see a bit of annelid structure. Can you see? They have the clitellum. So it's only something that is unique. Annelids. Right. And you can see annelids have a closed circulatory system. That means you can see the capillaries. Right. So only annelids have capillaries because they have a closed circulatory system. Clitellum is unique. Alright. What else? You can see the dorsal artery. Sorry, the blood vessel that is going. And this is the ventral nerve cord. Can you see? It says ventral vessel. It says I can't zoom in further. Right? It says ventral vessel. Can you see this yellow color? This yellow color thing that is running. That is a ventral nerve. That is common to arthropods. Except now. Right? But this ventral nerve cord you can see in arthropods as well. So the answer for that, the common feature is that. Okay. So this is the diagram for open circulatory system. Okay. Open circulatory system, right? So they don't have blood capillaries. What happens? They don't have blood vessels. They have hemolymph. Hemolymph is pumped into sinuses. Hemolymph is pumped into sinuses. And in those sinuses, the hemolymph surround the tissues and arterial exchange happens. So that is how circulatory system. Any questions from that MC? Anything you want to ask? Any questions? Moving on. Next question. Anemia is a deficiency syndrome of which of the following vitamins? Anemia is a deficiency symptom or syndrome of which of the following vitamins? Anemia. What do you mean by anemia? Usually reduced hemoglobin in the red blood cells. What are the vitamins that is given in your syllabus that are involved in hemoglobin production? Okay. So, if you are from ISM, I give you this leaflet that includes a short note. I mean, in that leaflet, on the back side of the leaflet, it contains the diagram of vitamin. It's the chart of vitamins, so you don't throw my handbill away, right? Okay, so if you refer that, you can see what vitamins the deficiency it causes anemia, then is definitely affecting involved in hemoglobin, right? Vitamin A deficiency is mainly related to skin health and vision, right? Skin health and vision. So you can't take vitamin A. Vitamin D is mainly related to bone health, right? Bone health. So you can't take. Thiamine is vitamin what? Vitamin B2. Thiamine. Yes. Not really. B2 is not involved in sort of hemoglobin production. It is sort of act as a, let's say, coenzyme and involved in sort of energy production reactions, right? So that's why reduced thiamine causes beriberi. Beriberi is involved with reduced energy levels, right? They can't produce enough ATP for their body functions. They are very weak. Beriberi are, right? Beriberi. Okay. So answer number 2, B12. B12. What is the name? Other name you can use for B12? Yes. Cobalamin. B12 is cobalamin. Right? So B12 cobalamin. Yes. Deficiency can cause anemia. B6. What is the name for B6? Pyridoxine. Right? Pyridoxine. It can cause anemia. Yes. What else? Give me 1 minute. Right. B6. Is it given in your syllabus? Yes. B6. Right. B6 pyridoxine. What are the deficiency symptoms? It's not given as in your syllabus as involved in. Yeah, that's why I have to double check, right? Uh, oh no, it's given, no? Yes, pyridoxine is involved in, yeah, it can cause anemia, right? So we can say folic acid is what? Between B9, folic acid, yes. You know, pregnant mothers, folic acid, right? Because that is to avoid the fetal anemia. Right? So I think answer number 2 is the best one. Right? Is it the answer you got? Yeah. Answer number 2. Okay. Vitamin K. Vitamin K is mainly related to what? Blood clotting. Mainly related to blood clotting. B1. What is B1? Do we talk about B1 in your syllabus? Yes, vitamin B2 is riboflavin. I'm very sorry. B1 is that. I never. B1 is thiamine, right? Okay. B1 thiamine causes this, right? So B1 here. B2 is riboflavin, right? Do I need to go through all the others, right? So the answer is vitamin B12, B6, and folic acid, right? So those are the. The deficiency can cause any. Deficiency can cause any. Okay. Which of the following statements regarding the respiratory system of man is correct, right? Respiratory system. Correct. Contraction of the intercostal. Forget about this external kappa, right? It's not in your syllabus, right? Intercostal muscles results in inspiration, right? What do you mean by what happens in inspiration? It's an active movement, right? What do we have to do? We have to increase the volume of the thoracic. We have to increase the volume of the thoracic cavity. So, how do we increase? We can do two things, right? So, our thoracic cavity has what we call as anteroposterior diaphragm. So, we can move it forward and upwards. We can do that, right? And also, our thoracic cavity and our abdomen is separated by what? Our diaphragm, right? So, we can increase what we call as vertical diaphragm. Vertical thoracic cavity is vertical height technology we can increase by. So this is our thoracic cavity, right? At rest the diaphragm is like this, right? So this is our vertical height of thoracic. So right, this is the head, this is the neck. So these are the trachea. Okay. So when you are breathing in, you can increase this diameter or the height by making the diaphragm more flat. It's not what they are asking here. They are asking if we take a look at it from this side. Right? So this is our neck. This is our chest. There's the diaphragm. We can increase the anteroposterior. Can sort of our the Islam sort of like what do you call it? That is called anteroposterior. Islam the vascular diameter. You can increase by the contraction of the intercostal muscle. So the moves like this because of what? There's a bone here that is called as the sternum. Right? There's nothing in the bone in sternum bone, right? That bone you can raise forward and also you can raise your ribs. How do you raise your ribs? By the contraction of intestines. Tracheal cavity is lined with columnar. Do we have columnar epithelium in the respiratory system? No. Yet we have usually is epithelium. Pseudostratified. Right? Pseudostratified epithelium and also ciliated. Pseudostratified is what we have. So it's right lung consists of two lobes, right? For this, let's talk a little bit of lung anatomy. Right? I think we are talking a lot about the lung. Right? So, pseudostratified, yes, it is. Yeah, I was thinking about this, right? It's a columnar epithelium. You can argue it is pseudostratified, but still it's columnar. You can see it's These are sort of elongated cells. Right? So what do you think? දෙකටම ඕනේ උත්තර. දෙකටම you need correct, right? Pseudo-certified ciliated column. Right. We can take. Right? I think I can give columnar epithelium as correct also. Right? What was it? Tracheal cavities lined with columnar epithelium. Yeah. We can also take it as, I mean, a correct answer. Yeah. Right. Right lung consists of two lobes. What do you think about the right lung? Right lung? It actually has how many lobes? Upper, middle, and lower it has three lobes. Okay. So right lung has three. The left lung has exchange of respiratory gases in the lung requires energy. No. Why? What is the method of respiratory gas exchange? Is it an active process? It's depending on the gas concentration or partial pressure difference. Partial. It's actually diffusion. In a primitive method, diffusion. Diffusion is a passive process, so you don't require. Final one, visceral pleura. Very, very nice statement. Visceral pleura adheres to the wall of the thoracic cavity. Visceral pleura adheres to the wall of the thoracic cavity. What do you mean by the visceral pleura? What are the pleura? Can you see this diagram? Can you see? Okay. What are the pleura? So I always say, imagine your lungs hitting, your lungs are, there's an immediate layer sort of pasted around the lungs. Right? There's an immediate layer pasted around the lungs. I will write in this blue color. Right? There's an immediate layer. No, there's a layer, or you can call it a plate, that is sort of pasted on the lung cells. Pasted. That is called what? This red color layer. Again, it's pasted on the lung parenchyma. And the lung tissue it's pasted. You can't separate. It's the visceral. And so your lungs are contained within your thoracic cavity. Thoracic cavity. There's another layer is attached to the thoracic cavity. Right? Can you see? No. Right. Anyway, right? That layer, can you see it starts from here? I'm drawing the blue layer, right? It starts from here and it is adhered to the inner surface of the thoracic cavity. That is called what? Parietal pleura. So, because of these two pleural layers, the parietal pleura, visceral pleura inside, the lung, when the thoracic cavity is expanding, the visceral pleura and the parietal pleura are also, they are free to slide together. They can slide. Free to slide together means what? They can smoothly move. Right? So they are also, these two pleura pasted to each other because there's a fluid. But we call that the pleural fluid. There's a fluid in between these two layers. The pleural fluid, have you heard of something called surface tension? Because of the surface tension, the two pleura are sort of they are connected or attached together, but they can slide faster. Because of that, when the thoracic cavity increases in size, the lungs also get bigger in size. Right? Clear about the pleural layers. The lungs are covered by visceral pavement. The inner side of the thoracic cavity is lined by the parietal pavement. In between them, you have a pleural fluid. Okay. So, what's this statement saying? Visceral pleura adheres to the wall of the thoracic cavity? No. Visceral pleura adheres to what? The lungs. What is the pleura attached to the thoracic cavity? The right. So there's a question in the resource. It says, the trachea, the respiratory epithelium is column, right? So that's what it's right. Respiratory epithelium is column. Yes, that's what the resource says, right? Sorry, it's the resource says the respiratory epithelium is pseudostratified. Yeah, that's what I also say. But it's pseudostratified column, right? I showed you earlier that. But so, I mean, if you want to take me one answer, the best answer is number one. Best answer is statement number one, yes. But answer number two is also not incorrect. Varadine. It's not wrong. Okay. Right. Statement number 30. Right. Incorrect statement regarding human blood. It's a specialized connective tissue. Connective tissues. Is blood a connective tissue? Blood can a connective tissue. Yes, it's a very specialized connective tissue. What are the specialized features of blood? Features of blood that makes it special. Blood. What are the features of blood that makes it very special? Things in a normal connective tissue. What are the things in a normal connective tissue? Let's say a typical connective tissue has to have what? Fibers, a matrix, and embedded cells. Those are the three components of a connective tissue. Typical connective tissue. You can't see the blue color. Right. You can't see the okay. Typical. Precisely. It has to have fibers, matrix, even cells, and matrix can be solid, semi-solid, or liquid. Right? So those are the variations. When we are moving to this is specialized because its fibers are not always there. Fibers are not always specific. Fibers are formed only in one situation. There is the one situation where fibers are formed in. What is the only situation fibers are formed in blood? Anyone? Only when blood is clotting. Can anyone tell me what is the fiber formed when we are clotting? What is the type of molecule that we have when blood is clotting? Fibrin. So that is one of the things that makes blood special. What is the other thing? Matrix is a liquid. No? Blood matrix is a liquid. What is the matrix of blood called? What is the special name for the matrix of blood? Blood plasma. Blood plasma is a liquid. So usually if you take connective tissue, areola, right, dense connective tissue, bone, cartilage, almost always it's solid, no? But it's a liquid here. Third one, cells are not produced by the tissue. Blood cells. How are blood cells produced? Most of the blood cells produced. Where do we have most of the blood cells? Blood cell production. Because in the bone marrow. So these are the features that make blood a specialized connective tissue. Right? Understood. So moving on to coming back to the MCQ. Yes, it is. It is a specialized connective tissue. A mature erythrocyte lacks of nucleus and mitochondria. What is the shape of a erythrocyte? If you take sort of a perihydrobial, it's like this. Biconvex lens shape. And if you draw it like this, it's like this. Biconvex lens. Sorry, I'm sorry. Yes. Concave, right? Biconcave. Sorry. Concave. Okay. Thank you. Right. That is, what is the purpose of having this shape in blood cells? What is the purpose? Anybody know? What is the purpose of having this shape? Increase surface area. That is the purpose. So that increases surface area because of the shape. Why doesn't it have nucleus and mitochondria? To increase the space available to have more hemoglobin. To increase the amount of hemoglobin. So yes, that is also true. Neutrophils and monocytes are leukocytes. What do you mean by leukocytes? White blood cells. Yes. Neutrophils and monocytes are white blood cells. Yes. There are 3 fields and 2 sites. No? What are the 3 fields and 2 sites? Basophils, eosinophils, and neutrophils. Clear? And what are the 2 sites? Monocytes, lymphocytes, and monocytes. All those are called leukocytes. Again, white blood cells. All those are called leukocytes or white blood cells. Most of the CO2 is transported in combination with hemoglobin. What are the ways that CO2 is transported? CO2 plus hemoglobin or minor hemoglobin. Right? CO2 can become combined with water and converted to, this is also reversible, it can become bicarbonate. HCO3 minus, right? So what are the percentages? 70% transferred, right? 23% is carbon and remaining 7% is free gas. Good. So most of the carbon dioxide is from this combination with hemoglobin transported and bicarbonate. So that is the correct. I mean that is the answer. Helps to regulate body temperature. Yes. Thermoregulation, there's a main part, right? The main role played by blood. Particularly if you can, I mean, when you are very hot, when you are in a very hot environment, have you seen your skin becomes red color? The skin becomes red. That is because the peripheral blood vessels are dilated and the blood is near your skin. Blood skin that means what? When the blood is near your skin, right? So when this is the skin surface now, when there's blood flowing under the skin, the heat is lost as radiation. Heat is lost as radiation. You can lose heat through blood. So it can regulate body temperature. You will learn about this under homeostasis. You will learn about this under homeostasis. Okay. Let's do this question and move on to do some essay question discussion. Okay. Which of the following is not part of the conducting system of the human? Not a part of the conducting system. Conducting system of the quick diagram. Conducting system of the heart enables the heart to be what? It enables the heart to beat independently. Can you remember? It enables the heart to beat independently. Because why? It can generate its own impulses. Even if a person is brain dead, the heart can keep on beating. Because the signal for the heartbeat is generated by the heart itself. The signal for the heartbeat is generated by the heart itself. That is fine. So always I tend to remember this as a hierarchy. Again, what do you mean by hierarchy? උඩින් කෙනෙක් ඉන්නවා යාර දෙවනියෙක් ඉන්නවා ඊළඟට එයාගේ සේවකයෝ ඉන්නවා එහෙම right. So in a country there's a president, there's a public minister, ඊළඟට there are ministers, then there are parliament ministers. So there's like a order of power. So in your heart, who is the person that is taking the decisions? Who's the person that is taking the decision? Sinoatrial node or the SA. Who is the deputy? Who is the deputy of the SA node? Who is the deputy of the SA node? Located here, the atrioceptor. We have what? AV node. AV. So starting from the AV node, we have the rest of the conducting system. What is the rest of the conducting system of the heart? We have the left and right. So before that we have the right. So we can say one of this, then left and right bundle branches. And this left and right branches in the ventricles, they divide into what? Yes, what is the final step? Purkinje fibers, right? Heavy bundle. This left, this right. Branches. And these out. This is the structure of the conducting system. So who is the what we call as the pacemaker? Who is I call as the person who is taking decisions? This one determines the beat, right? කොච්චර වේගෙන් beat වෙන්නේ and what is the rhythm? The speed and the rhythm is determined by this. That signal will be given to the AV. AV node transmits the rest. That decision taken by the SA node, the AV bundle, and to the left and right branches, and to the rest of the heart or the ventricles by the rest of the conducting system. Right? Okay? Clear? So what is the question asked? What is the question? Which of the following is not a part of the conducting system? AV node is a part of his SA node plugin times. AV bundle, you can call it as bundle of his net. AV bundle, we can call it as, right? I didn't mention here. AV bundle, we can call it as bundle of his. Coordinate entity. What do you know about coordinate entity? So I'll right. Where are you to find the coordinate entity? Where to find the coordinate entity? Right. So you know you have valves. There's the tricuspid valve here, and there's the bicuspid valve here. These valves anchor to this what we call as the papillary muscles. These papillary muscles are anchored to these papillary muscles, and they are connected to those papillary muscles by coordinate tendons. Coordinate tendons. Fibrous tendons giving support to the. I mean, they prevent the valves from being turned inside out. Right. So these are coordinated. Coordinated. Clear? So what is the part conducting system? Coordinated. Alright. Let's move on. I mean, okay, let's do one more MCQ and move to some remaining essay question. During the opening of stomatical open, sodium ions are actively transported. Is it sodium ions? What do we call this sort of process? It's called K plus influx, right? Stomata arrangement. Hypothesis 7, according to what hypothesis? So stomata composed of two guards, they are sort of like this, right? Remember there are cellulose bands, right? There are cellulose bands. Stomatal opening happens because of what? Potassium ions are pumping into the guard cells, and therefore water also comes inside, right? Water also comes in because of the decreased water potential. So the stomata, right? That's how it is. So it is not sodium ions, it is Kx. During opening, the turgor pressure of the guard cell decreases. No, because the water is flowing in. Any lag you can clearly hear. Let's see. Issues. Can you hear me correctly? Clearly? Yes. Okay. Right. So, what did I say? Because the water potential is decreasing because of this K plus influx, water comes in. When water comes in, the guard cell swells. Swelling means what? There is increase in turgor pressure, not decrease. Right? Carbon dioxide content in the substomatal cavity increases. So, what are we talking about there? Substomatal cavity. What do you mean by substomatal cavity? So, if this is the lower epidermis of the leaf, right? So, these are the guard cells. So, this is the stomach. Above that, what do we have? Mesophyll cells, right? Mesophyll cells in. So, this is the substomatal cavity. So, stomata has to open, or the guard cells have to open, because why? Why do we need robots? Do you plant cells? No. Why do you plant cells? To take dioxide in. To take carbon dioxide in. To fix. Right? For the fixation. Fixation. Fixed by photosynthesis, carbon dioxide is fixed. Yeah. So stomata, when inside the substomatal cavity, when the CO2 concentration increases, the stomata doesn't need to open because there is enough carbon dioxide. So, no. When the CO2 concentration increases, no need for the stomata. You understand? During the opening, water potential decreases. Why does the water potential decrease? Because of this K plus influx. K plus influx. So that is the correct answer. Potassium ions are passively transported. This is an active process. This is an active process. It is powered by what? It is powered by photosynthesis. Because guard cells are green. These guard cells are photosynthetic. Guard cells are photosynthetic. Therefore, they can produce energy to transport the potassium ions. Got it? Next one. Which of the following statement is incorrect regarding phloem transport? Incorrect regarding phloem transport. They are asking the incorrect statement. What is the hypothesis that we use to explain phloem transport? Pressure flow hypothesis. Pressure flow. Pressure flow. Technology pressure flow hypothesis. What happens in pressure flow hypothesis? So there's a phloem vessel like this. Composed of what? CO tube elements. Phloem vessel. Phloem flows from where to where? Phloem flows from the source to a sink. This is the direction. What happens in this process? Sugar is loaded in the near the source. Because of this sugar loading, what is going to happen? The water potential decreases. So there's a nearby xylem. What does xylem have? Water. So because the water potential decreases in the phloem, water in the xylem comes into the phloem near the source. So I always tell to imagine phloem movement like squeezing a syringe. Squeezing a syringe. Right? It's like squeezing a syringe. What happens when you squeeze a syringe? The pressure increases, therefore the liquid moves. That's what happens. So here, near the source, pressure increases, therefore phloem moves from source to side. Pressure on. Incorrect. Translocation of phloem sap from leaves to roots takes place through apoplast of CO tube elements. What do you think? Can we consider phloem movement as apoplast? Can you see from one MCQ I'm covering a lot of theory. Right? From one MCQ I'm covering a lot of theory. Right? So what is the definition of apoplast? What is the definition of apoplast? Movement of something through the movement of dead cells. Apoplastic pathway is always a movement through dead cells. Okay? It's always a movement through dead cells. Okay? So, CO tube elements. So, phloem translocation takes place through CO tube elements. Are CO tube elements dead cells? Are they dead cells? No, right? So, can we consider phloem translocation as an apoplast? Because why? It occurs through living cells, so we can't consider it as an apoplast. Mature leaves are the major sugar sources. Yes. Because why? They are photosynthesized. They are the main sources of photosynthesis. Sorry, they are the main places of photosynthesis. So they produce the most amount of sugar. Developing root epicels and shoot epicels are sugar sinks. Right. Anywhere where there is development, it's going to consume sugar. It requires energy. So these are usual sugar sinks. This phloem unloading. So loading and unloading are active processes. What do you think? Phloem loading and actually, if you take this phloem loading, so this is loading, sugar loading happens here. Actually, sugar loading is active. Sugar loading is active because why? In the phloem, I mean the CO2, the sugar concentration is high. So from the source to the phloem, you have to actively transport the sugar. But what happens in the sink? In the sink, sugar concentration is low. Because why? The sink is using the sugar. The sink is using the sugar. So usually here, the sugar unloading is passive. I always tell to imagine like so. Imagine you for some construction you order some sand. Yeah, very lorry again, very load order some a lorry of sand. Right? So how do you load that lorry? Using hand, using shovels, you have to load the shovel. But when the lorry comes to your home, they are in the construction. How do you unload the sand? You can sort of tip a ground, right? Tip it. Because why? Here, near the source, the phloem or the CU tube has high sugar. So you have to actively load. In the sink, the sugar is being used. So the sugar in the phloem or the CO tube can come easily. That's why we say. Usually phloem loading is active, unloading is passive. So we can say, you can say this also as well. Right? There can be more than translocation of phloem sap to one CO tube element to the next is a passive process. Right? You can also this one you can also take as wrong because why? The phloem loading is an active process. Therefore, it requires energy, so you cannot consider it as passive. So don't think too much about what's the answers. Right? These are questions. I'm using these questions in order to revise. And mind you of the theory. Right? It doesn't matter. You can have several. You can argue with me if something you think something is wrong. You can argue with me and say why not like this. So the point is, you have to think logically when you are arriving at the answer. Okay. Let's quickly move on to the next question. The blood group that can be received by a person that B minus during a B negative during a blood. What is the logic behind blood transfusion? I'm not going to teach this logic, right? So full theory, right? What is logic behind that pronunciation? Person with B means they have B antigen. B antigen. B negative means they do not have the rhesus. RH negative, right? Correct. So a person with blood group B minus. So the logic I like to tell these people is, I mean when I teach is, so here's the patient. I may have patient here, right? So what is his blood group? B minus. We have to give him blood. With accident, he has he is having dehydration, probably mainly it's bleeding, hemorrhaging. We have to give him, or maybe he's severe anemia, severely low hemoglobin. We have to give him. So his blood group is B minus. So these are his blood cells. What is going to happen if we give blood having an antigen that his body doesn't know? That will cause a reaction. So what are the antigens that his body knows? Yeah, then the double antigens would only B. The only antigen his body knows is B. What are the antigens his body doesn't know? Yeah, body ka danna නැති antigen. His body doesn't know A antigen. Yeah, danne. His body doesn't know about A antigen. His body doesn't know also the rhesus because why is B negative. So the logic is, if you give someone blood having an antigen he doesn't know, that is going to cause the problem is called agglutination. Can cause agglutination. That means the blood you give will be destroyed by the person receiving. Will be destroyed by the person receiving. So what are the blood groups that you can give him? Obviously, you can give his own blood. You can give him the same blood as his. What is the blood that doesn't have any antigen? किसी में antigen नहीं है blood. What is a blood group that doesn't have any antigen? Yes, I need answers. There is a blood group that doesn't have any antigen. Red blood cell लेके किसी में antigen नहीं कहते कोई blood. Yes, blood group O. O doesn't have any antigen, but you cannot give any RH. So you can give O negative, right? You cannot give rhesus containing. What is the answer? B negative, B1, B negative, and all. Right. Clear? Any questions? Any questions? We have, I think, about 6 more MCQs, right? No, 7, about 7, right? But I want to discuss this essay. Describe the histological structure of the liver. Right? Describe the histological structure of the liver. What do you have? The histology of the liver. What do you mean by histology? That is the cellular arrangement at a tissue level. At a tissue level, how cells are arranged. If you take the liver, the liver is like a let's say a month protein, like industrial zones, The liver is like that. It's like an industrial zone. It's like a collection of so many factories. A collection of so many factories and filtration plants. We have water purifying plants again. The liver is like a filtration plant. It's like a factory, right? So many things are right. So the liver is like a collection of these factories. What do you call one of these single factories? We can call functional unit of the liver. What is the functional unit of the liver? It's a hexagonal shaped structure. It's a hexagonal shaped structure. Yes, alright. Yes, anyone? It's a hexagonal shaped structure called the liver. Anyone? Liver lobule. It's a hexagonal shaped structure called the liver lobule. Right? So, what are the things you have to know about the liver lobule? What are the things you have to know about the liver lobule? At the center of a liver lobule, you have a vein. At the center, you have a vein that is called the central vein. That is taking blood away from the liver towards the heart. Taking blood away from the liver towards the heart. Right? And then radiating from the central vein, we have liver cells. What do you call liver cells? Hepatocytes. Hepatocytes. Hepato means anything to do with the liver. The liver එක සම්මත වුණ දෙයකට කියන්නේ hep. Right? Hepatitis. Right? Liver එක ලොකු වෙනකොට අපි කියනවා hepatomegaly. Liver cells are hepatocytes. These hepatocytes are arranged as columns. They are arranged as pairs of columns. Can you see? These are liver cells. Right? These squares. No? These are liver cells. They are called hepatocytes. These hepatocytes are pairs. Hepatocytes are pairs of. Right? You have to be able to draw this. Always, you only draw something if you have to be able to. Okay, you know this, right? I'm not a big fan of drawing diagrams. So if I draw something in live terms, that means in the exam they can ask you to draw this, and they will give this, right? So these are what? These are pairs of columns of liver cells. In between liver cells, right? In between these pairs of columns, we have blood vessels, but they are complete blood vessels. They are not complete blood vessels. They are very leaky blood vessels. They are very leaky blood vessels. They are called what? These leaky blood vessels are called what? Anyone? What are these leaky blood vessels called? They are called sinusoids, right? Sinusoids. And also, in between, two columns, we have passages for bile. This bile is produced from the liver. Bile is produced by the liver. Bile contains what? Bile salts and bile pigments. So there are channels for the bile to flow as well. These are bile canalicular canals for bile. Bile canals for bile. In between columns, bile canalicular. What else? Then there are some special white blood cells. There are some special white blood cells on the edges of these hepatocytes. They are called hepatic macrophages. Hepatic macrophages are lymphocytic cells. They have a special name. What is the special name? What is their special name? Can you give any special name for these given these hepatic macrophages? Anyone? You can send to me private. For cells. For cells. The labeling. For cells. So what about the vessels? So we drew one vein that is in the center. What are the other vessels? Actually, the liver, it's like a if you go to the back of a computer, you can see many wires connected to the computer. So the liver is like there's three cords, right? Power cords, three main blood vessels that are connected to the liver. Three main blood vessels, right? Drinking blood, you can't see blue. Yeah. Two blood vessels bringing blood to the two blood vessels bringing blood. That is the hepatic artery, hepatic portal. Then taking blood away. Blood away from vein. Vein එකක් සාමාන්‍ය blood takes blood away from the organ towards the right. So these are the blood vessels that are coming and going from the liver. Okay. So we have drawn one. So where are the others? Where are the other blood vessels connected? I draw a bunch of hepatic. Almost every corner we have a branch of the hepatic. And also you have a branch of the hepatic portal vein. I'll draw it in this blue color. Yes. And at the same time, in each corner, there's a duct to take away the bile. මේ bile කැනලිතුමේ දිගේ එන right? Bile කැනලිතුමේ දිගේ එන bile. To take that bile away, you have what? Yes, the bile duct. These are the things, right? So let's label. What is this? Branch of hepatic portal vein. This is hepatic. This is bile duct. It's a branch of the bile. So those are the branches. These are what? Sinusoids. These are what? Upper cells. Upper cells. These are what? Colony dot. Hepatocells. Columns. These are what? This green color. I label here. Fine. I can. Okay. This is the liver lobule that you have to be able to find. And you have to be able to describe, and this is the branch of central vein. Or you can say central vein is a branch of the hepatic vein. Okay, clear. So let's mark this. Other things you have mentioned, histological structure of the liver. I think it's yeah, so this is the respiratory system. Yes. So I'll just show you this for your just interest, right? How the back of the liver looks like. Back of the liver looks like, right? How the back of the liver looks like. So you can see the different arts, right? So this is the gallbladder, right? This is the gallbladder, right? This is the bile duct, okay? And this is the portal vein, right? This is the portal, right? This is the hepatic artery. Can you see the red color? And this other blue one is the hepatic vein. Two veins. The vein and just for your interest. This sort of the vein. Right? So okay. Histological structure of the liver. What do you have to say? You have to start with here. Just a small intro. Liver is the largest gland. I like if you that. Each, I mean, when you say liver contains four lobes, that you have to say. Each lobe is made up of many hexagon-shaped lobules, which are the functional units. Another lobules are made of cuboidal cells called hepatocytes, which are arranged in pairs of columns from a radiating from the central vein. Between two pairs, very important, there are sinusoids containing a mixture of blood from the tiny branches of the portal vein and the hepatic vein. These are two pairs. This arrangement allows venous blood, high concentration of nutritional materials, to mix with venous blood, to mix with arterial blood, and come into contact with juice. Hepatic macrophages, next point, are found in the lining. Blood drains from the sinusoids into the central vein. Eventually, the central vein is joined from the hepatic. Bile canaliculi run between the columns. Canaliculi join up to form larger bile canals. In the corner of hexagonal structure, branch of hepatic artery, portal vein, interlobular. What do you mean by interlobular? Among lobules, right? Interlobular bile duct. That is it, right? Those are the histological structure of the liver. Then, function of the liver related to digestion, that is part 2. Liver secretes bile stored in the gallbladder until released into the stool. Bile contains bile salts, which act as emulsifiers, helping in filter fat digestion and absorption. Yes. Most of the absorbed nutrients reach the liver. It regulates the distribution of nutrients to the rest. excess glucose is stored as glycogen. Glycogen is a key. Glycogen deposition and breakdown is regulated by insulin and glucagon. Fat soluble vitamins, you have to mention all, CADE, and some water soluble vitamins such as B12, iron, copper, are stored in the liver. Got it? So that is that is. So I think that is enough for today's discussion. For the rest of the MCQs and the remaining essay question, I will send the marking scheme. Right. So as a summary for people who are watching the recording as well as join live. This is the start of our paper program. 10 month, just now only 9 months and maybe week remaining. So this is the first of our paper. Okay. So as you can see, you are just answering, you joining with the active recall session. Active recall. Right? We are doing questions and we are rapidly revising the theory part as well. So that's the importance of doing questions. Because in bio, if you only just read the results and try to memorize, that's not going to be effective in the long run, right? That's why this sort of active methods, right, doing questions and recalling the theory is important. So I'm going to guide you through that process in this 9 months. So that's the plan of this paper program. Okay. Any questions that you want to clarify? Any questions? From anything that we did today? If you want to clarify? The paper discussions it's sort of going to be sort of once a month situation. So we are going to do this sort of long paper discussions once. So that will involve, I will let you know the date, I will put a poll and decide on a proper date, right? Could be Saturday, thinking of Saturday evening, the paper class. For this, I sort of, because before the end of this month, I have to do a paper discussion. That's why I Tuesday as the date for this discussion. But that will depend. So probably Saturday will be. Okay. Anyone else? Any question?
Recording stopped.
Yeah, I'm starting support a moment next month. This next month, probably second week. That's going to be a special bonus semi, right? With if you can come to ISM, you can have a hands-on session. Okay. Any other question? Okay, then we can finish for the day.